So good afternoon, everybody, and welcome to this first webinar from the first series of VALITEST webinars. So this series will be on the concept of test validation in plant health. And the objective of the series are to introduce the concept of validation and present the state of the art in terms of choice of the test and the analysis of the results of performance evaluation, and to train experts on the use on kits, including on-site and field tests. So today's webinar will be the first one of the series. It will be about what is test validation and why it matters for a reliable diagnostics. So the webinar will be presented by Françoise Peter from EPPO. So Françoise Peter is the deputy head of the European and Mediterranean Plant Protection Organization of EPPO. And so she will make the presentation today. So it was supposed to be a duo between Françoise Peter and René van der Vlugt. But unfortunately, René van der Vlugt was not able to join today. So the presentation will be all made by Françoise Peter. So Françoise, if you can go to the next slide. So as you can see, this presentation is the first one of a round of 5 webinars that will be held in this series. And in this series, we will also have 2 practical training sessions. So one on the concept of validation and one on the use of kits and on the use of kits. So Françoise, I don't know why, but we can see 2 windows.
Okay. So what is it? Well, because we changed the room because of the drilling, so I wasn't sure I had the, yeah, I didn't project the correct the one because I was disconnected from the network, and so I'm gonna, if you allow
you can
me
switch
for 2 seconds.
presentation.
I'm sorry for the attendees, but we have serious drilling going on just above my room, so we had to do an emergency change of location. So I'm gonna put the correct presentation now. This one I have to escape, and now close, and the other one is the correct one. Apologies for that. So René is mentioned, and okay, now you can continue.
So no, yeah, I was done with my introduction. So there will be 5 webinars in the series. And of course, you can register for all those webinars. So the first one today is the general webinar on validation. The second one will be more focused on how to adopt a new test in your lab. The third one will be focused on on-site tests. The fourth one will be more on how companies that are making kits and the quality control and validation of their products, and how the European Plant and Diagnostic Industry Association Charter will help to improve this test. And the last webinar will be on communication on test selection between risk managers and diagnostic labs. So I will give you the floor, Françoise. And,
thank you very much, Charlotte. And yes, of course, I regret that René could not do the presentation, and so I didn't see any drop when you said it's in the attendance. So, but so I will try to guide you through this first webinar of VALITEST, and we will start first with a first question, and Charlotte, so people can answer. So you have a poll, and you can answer the first question, which is, are you familiar with the concept of test validation? So you have the possibility to indicate in the poll what How many people are familiar? So we have 90% of votes for the moment. It seems that it remains pretty much stable at 90%. So we might consider that we have a good view about the people who are present in this webinar. So we have a majority of people who are familiar with the theoretical concept, but not on the detail. I suspect that's the answer, Charlotte. And we have 30% of people who've already validated several tests and 15% who do not know the concept. So it's an introduction webinar. So of course, many of the people will already be familiar with what I'm gonna show you during this presentation. But let's go. So first, so the presentation will be in 4 parts. So the first part is about the concept of the regulatory plant-based diagnostic and why we need accurate and reliable tests. The second one will be the choice of a test or combination of tests, depends on the circumstances of use. So we will discuss this a little bit. Then we have the how validation is part of quality assurance and essential for accreditation, to finish with how is a test validated. So why do we need accurate diagnostic, and what do we do in plant health that is based on diagnostics? The National Plant Protection Organization have to organize surveillance and to delimit the distribution of known pests, but also to detect new ones. When a pest outbreak occurs, then NPPOs have also to manage outbreaks. So that's for the work of the NPPOs. They are also in charge of import and export inspections, where they do take samples at import to do analysis. And again, the same is true for export. So when you have to certify a consignment for export, you do inspection of consignment to issue the phytosanitary certificate. And it's also when doing this export inspection that a diagnostic laboratory analyses are conducted. So a second part where we need. So for all this, we need the laboratory analysis in the inspection that are done to detect and identify pests. But there is also another activity which is important, which is the certification of plants in the framework of plant propagation material. And you need testing at the different propagation stage to demonstrate, for example, that your material is virus free. And so your testing is performed at different moments during the production of your propagating material. And also you also have some time there are also tolerance level in this certification scheme that you need to control, and you can do this with testing. But then what is really essential to understand, and you all know it if you're part of the regulatory diagnostic, is that a test result may have consequences. And the consequence is, for example, that there will be a phytosanitary action taken. And then for the producers, for the importer, for the exporter, this is really where there can be consequences. And they can be financial, but as you can see on this picture, you could also have social and environmental consequences. So I've taken one example from Europe, which is the eradication of Xylella fastidiosa in an almond orchard in Alicante, where there was, so eradication has been required and there were social consequences there. And the other example is HLB in citrus groves in Florida, where you can see that, yeah, this has consequences where you have to destroy an entire orchard. You also have the consignments at import and at export that you can reject or destroy. And also in this case, you have an element which is true in the other cases as well, but particularly true there, is that you really need the speed of diagnostics. So the consignment cannot stay for weeks waiting for a result. You really need to have a quick result, so which is an additional element. So basically, we need rapid and accurate diagnostic, and this diagnostic has consequences. And so this is why you need to be sure or to have as much reliability as possible in your diagnostic. So the choice of a test or a combination of tests depends on the circumstances of use. Indeed, I've already mentioned some of them, but the diagnostic tests are used in many different circumstances. You may need to test as part of a routine survey for a pest which is widely established in a country. That's one case. But you also have to do surveillance to demonstrate a pest status. So, for example, demonstrate that the pest is not present in your territory. You also have to do surveillance for pests you don't see, so latent infestation. And in this case, there's no choice but testing. Surveillance, part of the official control or part of an eradication program. You've seen some examples before. The phytosanitary certification at export or the propagation material, again, an example that I've given already. Imported consignment need to be tested as well. And also an important point is to detect a pest in an area where it's not known to occur. And finally, there is also one case, a circumstance is that the detection of a pest in a consignment that is originating in a country where it's not supposed to be there. So depending on all these different circumstances, you need different test characteristics, and you also see different level of reliability that need to be achieved with your test result. So the first thing I like to start with usually, because you will hear me talking a lot about a test. So I think it's important that to start with, I explain what we in EPPO has been agreed in terms of terminology. So assay, test, method, protocol, diagnostic protocol are kind of equivalent. But in EPPO, what we have here is a test which is equivalent to an assay and which is equivalent to a protocol. Although in recent discussion, the equivalence is more on test and assay for sure. So a test for us, and that's what you need to remember, is a combination of a method, a target pest, and a matrix. So that's how a test is qualified. And when we speak about protocol, EPPO diagnostic protocol, it's usually a combination of tests that we are talking about. So the validation of tests. So clearly, if you are validating, you are doing this because you also follow a quality assurance program, and for many labs you are accredited. So to start with, it is really essential that the laboratory work under quality assurance, and validation of tests is done under quality assurance. So the quality systems, you certainly all know about it. They are the system that ensure the quality and the confidence in the laboratory provided by a laboratory, in the results, provided by a laboratory. So that's basically what we aim with quality system. And you have what we call clients in the quality system, and you need to ensure that what you deliver to your client is what he needs. So the second question we have now is about the quality assurance system. So who is in the audience working under a quality assurance system? Voting progressing. We've nearly reached the same level than for the first question. So I think we can. So we have 80%, 20% roughly. Okay. So the very kind of early on in the organization, in EPPO, we have considered that minimum general quality requirements were needed to perform plant-based diagnostics. And these have been defined and agreed in a standard, which is our stand PM784, standard on basic requirements for quality assurance. And this standard was approved in 2007 and revised in 2018. So what does it cover? In fact, it tells you that in order to work under quality, there are a number of points that you have to ensure in your laboratory. So first, this is a management system. And so that covers your facilities and also the activities of the organization. And what does this mean? It means that you should have the resources available to carry your activity. And that's about the personnel, that's about the facilities, that's about the consumables. So the management system should ensure that you have this in your lab. You should also have clear responsibilities and tasks of your staff defined. You should have standards operating procedure available. So all the instruction to work in the lab to perform your diagnostic should be available. You should have requirements regarding confidentiality and potential conflicts of interest. You should establish internal audits in your lab, and you should have a documentation system in place. There are also technical requirements. So the fact that you have the staff, sufficient staff, but you should also have qualified personnel. So that means their basic training, but also that they have to be trained whenever needed. So it's a continuous training, but in addition to the basic qualification. You should also be able to select the plant-based diagnostic test in an appropriate way. You should have appropriate equipment, reference material, cultures, or access to sampling, if you do sampling, or how you handle the sample, all that should be described, and also how you manage your control, in when you perform your testing. And also you should follow some rules regarding the reporting of the results. The second step after the basic quality system is accreditation. And so in the EPPO region, the accreditation of EPPO laboratories, not EPPO laboratories, we don't have any laboratories, but the laboratories in the EPPO region are according to the ISO standard 17025. And it's worth noting that for some laboratories, in particular the labs in the EPPO region, this is a legislative requirement. And so, but what an important point is that accreditation is given by national accreditation bodies. So each country has a national accreditation body, to tell the story short, and the accreditation is granted by this accreditation body. So the standard ISO 17025 exists for some years now, and we started in EPPO by thinking that this standard was enough, but in the years 2004-5, we started the expert asked us to ensure through a standard that we can achieve a better harmonization of the interpretation of this standard for plant-based diagnostic. And this is when we approved the standard, and it's a new number, PM798, which was approved in 2009, and this gives you an interpretation specific for plant-based diagnostics. But important point is that it includes guidance per discipline on how to perform the validation of a test, and this standard was approved in 2009, but was revised already 3 times and is about to be revised a 4th time. And why do I speak about this standard more specifically? It's because it gives a guidance, and it was also one of the standards taken as a basis in VALITEST to do the work done in the project VALITEST. So, third question, how does your laboratory perform tests under ISO 17025 accreditation? Okay, so we've reached again the 90%. So we are this time on quality assurance, we were 80% yes, 20% roughly no. Now we are 63-37. So on accreditation, we have less labs in the audience working under accreditation. So that takes us to the core of this talk. How is a test validated? So when you are a laboratory preparing for accreditation, you should only use validated tests. And how do you do this validation? By evaluating the test performance characteristics. And there are different performance criteria used to characterize the test. And the most important, those that have been considered as the most important by the EPPO experts are those that are mentioned here. Analytical sensitivity, which is how much. Analytical specificity, which is what. Reproducibility, so does the fact that I change the operator, the time of I do the analysis or the equipment change the result? Repeatability, do I get the same result between replicates? And depending on the scope, you look at the selectivity. So what will the variation of my matrix, will it impact the test? So before I go deeper into validation, there is an important point in which I need to explain the difference between validation and verification. And this is also why we went through that standard and explained everything about validation, because we wanted really labs to understand what they needed to do when they want to implement a test in their lab. So if the test has validation data already, and you will see later that we also analyze this validation data, then the laboratory will, I would say only, but will need to confirm its competence in performing the test. And there is a process described in our standard for that. On the other hand, if there is no or incomplete validation data, then the laboratory should produce the missing validation data. And when you take a test from the literature, you will see, and you have seen, because you're all in labs much more than I am, you will see that in the publication, you don't always find all the validation data that you need. So you will need to look at what's available and then produce yourself the missing validation data or the partly missing validation data. So again, we should first, before we go on how really we do it, we should look a little bit into what these criteria are. So I said how much. So analytical sensitivity, so how much, so is defined as the smallest amount of target that can be detected reliably. And this is sometimes referred to as the limit of detection. So you might be more familiar with limit of detection. And we provide further detail on how to do this in the standard PM798. The analytical specificity is divided in two parts. The inclusivity, so do I catch with my test all what I need to catch? So in fact, it's the performance of a test with a range of target organisms covering genetic diversity, different geographical origin and host. So you look into the variability of your target and you ensure that the test will catch it. On the contrary, the exclusivity is how your test will perform with regard to cross-reaction with a range of non-targets. And this could be closely related organism or organisms that are present on the plant you are testing. So what you're trying to do here is to ensure that you will not pick the wrong organism. So that's the exclusivity. Then you also look at the repeatability again. So the replicates. So do I get the same? Do I have, this is to read the definition, the level of agreement between replicates of a sample tested under the same condition. And the reproducibility is the ability of a test to provide consistent results when applied to aliquots of the same sample tested under different conditions. So if you can, you vary the person, you vary the time, the equipment, and if you are lucky to have several locations in one entity, you can also vary the location. The selectivity is the effect of the matrix to the test performance. And then there are two more criteria, which are the diagnostic sensitivity and the diagnostic specificity. And then in this case, you are comparing your test with another test, or and so this gives you what is often known as the proportion of true positive compared to the true positive plus the false negative. And so, and you have the diagnostic specificity, which is again a proportion of uninfected, the true negative compared to the true negative plus the false positives. So, but the first criteria, the last two one, are not criteria that we require, for example, on a systematic basis in our diagnostic protocols, for example. But if we have the information, of course, this is additional information which is valuable. So, so you will tell me, okay, so these are the performance criteria that I need to look at. So that's some work to be done. So how do I tackle this work? So the first step is to consider which performance criteria need to be evaluated based on a risk analysis. So to do that, the risk analysis will look at first the context of use of your test. And so do you intend to use your test? So first, you should define the scope. And the scope is the organism, the matrix, and the method. And then will I use the test as a screening test, as an on-site testing, as a confirmation test? Does the client have specific needs? For example, they want a specific level of analytical sensitivity. What will be the impact of the test results? So do I need more reliability in my test? Because I know there will be the impact is important. What do I need for the level of confidence? If I have a pest that has a very large host range, can I define the number of hosts on which I will do the evaluation rather than trying to be so very exhaustive? Do I have specific requirements for specific strains to be detected? So it always come to my mind an example of a nematodes where there was validation data, but a specific species which was present in one region and not in the other part of the region was posing problems. So for that part, it was important to be able to distinguish from that specific species present in one part of the region. And so is this evaluation of possible cross-reaction? Do we need some, do you need some flexibility in terms of, for example, equipment, etc.? So these are some elements that are described and need to be can be considered in the context of use. Then the second step will be to define adequate performance characteristics. So what do you expect from the test? And then to review the validation or other data already available. And then you look at the possible altered condition, which is, for example, will all these reagents or chemicals be sustainably available? So can I get this reagent on a long term? What happens if I change one of the reagents? What happens if I change the equipment? So this is the elements you then consider. And then the outcome of this risk analysis will tell you what you need to do next. So maybe the test you envisage is not appropriate and you need to select another one or develop a new test. But if your conclusion is positive, then your conclusion could be that you need you need to produce the performance characteristic of the test because they are not available. You could see that some performance characteristics are available. So for example, you work on the basis of an article, and the article has really done a lot of work on the analytical sensitivity, but on the analytical specificity, there is a need to complete the evaluation with more species, more lookalike. Then in this case, you will produce the validation data for that performance criteria, but then do a verification for the other performance criteria. And if all the relevant performance characteristics are available, then you can go for the verification process. So there are some examples of outputs of risk analysis. So you could consider that if data from a test using the same method can be transferred from one test to another, you could limit the extent of validation for selectivity, repeatability, and reproducibility. And an example in the region is that experience with real-time PCR for flavescence dorée allows the extent of validation for repeatability and reproducibility for bois noir to be reduced, for example. Regarding analytical sensitivity, if the test is only used for example, isolated specimens, the quantity of your target is in this case, for example, if we're talking molecular test, is not a limiting factor. And then you can reduce the extent of validation for analytical sensitivity if you always use the test in isolated specimen or specimen. But what you should check is a potential inhibition effect. So, but that's again an example where you would not need to do all the different steps for analytical sensitivity. And for for analytical specificity, you don't need in some cases to be specific, or if you want just to use the test as a generic test for example, post-pivaroids, you don't need to go into the analytical specificity to the same extent as you would do for other tests. And again, I will come back to that earlier. This is because usually you're never relying on a single test. So how to generate the performance characteristics? So again, I'm sorry I talk a lot about the EPPO standards there, but these are guidance that have been established together with the experts from the EPPO region, and so they agreed that this was the way to perform the validation and generate the performance characteristics. So in this PM 798, you will find annexes providing recommendation for the evaluation of the performance criteria per discipline and for different methods. So here you have the examples of molecular. So you have a copy of the table for bacteriology and molecular methods. And basically what it says is the number for the different, again, methods, you will find guidance on the number of experiments you can do, how you do them. So here they use spiked sample with extract with a range of cells of the target bacterium per ml. And so this is giving you the detailed guidance on how practically you can do the evaluation of sensitivity. Here again, for analytical specificity, it tells you that you need to analyze a range of the target strains of the target bacterium. So you do your analysis of what you want to achieve, and also what you can encounter in your country, for example, if it is in surveillance, and then you select the strains you want to evaluate. And the same for the non-target bacteria. You select a number of strains that you want to, the number of bacteria that you want to evaluate in your exclusivity testing. And then for them, you will then evaluate them during the validation process. So repeatability, again, is based on the analysis of 3 replicates of some spike sample extract. And then you do this at low concentration to ensure that you will get a good picture of your repeatability. And for the reproducibility, you do the same as for repeatability, but you change the operators, you do it on different days and different equipment. And selectivity, you will use different, for example, cultivars of the host plant to see how it affects the test performance. So just to inform you that we wanted in EPPO to encourage collaboration, communication between labs. And this is why we've established first a database on diagnostic expertise, where you can find expertise of experts working in labs for regulatory diagnostics. And in this database, you also find information provided by laboratories or generated in the framework of projects regarding validation. And so I can only encourage you to go and check the list of the validation data. So this is still work in progress. We are gradually transferring the data from the previous database. So the database was completely redone through in the framework of the VALITEST project. In particular, this section on validation, which is now much more searchable than in the past, but the previous validation data, which could only be generated as PDF format, is now gradually being transferred in the new, more searchable database. So, but one important question is how to use the validation data to decide on the test to use. And I will only give some examples because there is a practical session on the 26th of January. Unfortunately, the registration is closed, but it's specifically about analysis of performance characteristic. And this is really to answer this question, but I will give you some few examples. And I won't start with the easiest pest in the world, for sure. Xylella fastidiosa is showing a little bit the extreme of the difficulty of validation. So you have an organism that has subspecies, that has vectors, that has more than, I should change the figure to 500 hosts. With a very big number of hosts. And I don't know if you remember, but during my presentation, I said sometimes you need to prioritize the host on which you will do your validation. Xylella is a really good example of that. You cannot validate on all the potential hosts that are hosts of Xylella fastidiosa. So Xylella fastidiosa is also a good example on which test is considered suitable and which test is not considered suitable, and can it be considered suitable in some situation. So for example, in the screening tests that are in the EPPO diagnostic protocol for Xylella fastidiosa, you have serological tests and PCR tests. And clearly, when you are in asymptomatic plants from a pest-free area, Xylella is not the test that should be used. So in this case, you really want a test that is more sensitive, a lower analytical sensitivity. So in our diagnostic protocol for Xylella fastidiosa, serological tests are can be used for symptomatic plants from a known outbreak or a buffer zone around an outbreak. So that's an area where you can use ELISA to detect the bacteria. Another element is that for some of the first tests you do as screening test, you can accept a lack of analytical specificity for the screening test because you will do another test after. And this is really an important point to remember. It's rarely the case that only one test is used to give a positive result. There is a confirmation, and then you can accept to use a less specific test as a first test, but then and a more sensitive one as a first test, but then have a less sensitive one as a second test. And in particular, for example, for bacteria, that's not the case for Xylella, which is not very easy to grow, to culture. But in some case, if you have isolation, then you can have a positive, for example, a positive test on plant extract, then isolation, and then a less sensitive test on your culture. As we explained, the quantity will not be a problem. Another point that is really clear with clearly illustrated with Xylella is that the analytical sensitivity may vary depending on the matrix, and also and that's valid not only for Xylella, but that the nucleic acid extraction is critical, and different nucleic acid extraction methods may result in different analytical sensitivity. So that's also an important point for validation. Another example is Candidatus, another diagnostic protocol. So on Candidatus Phytoplasma, Mali, Piri, and Prunorum. So where you have a clear indication that, for example, when you're dealing with imported plant material versus material tested for specific phytoplasma survey, it's useful to perform a generic test. And then after that, you go for the specific test. And it's also the same. You can often find this, that you use a generic test first and then you go for the more specific test after. It's also a way of not missing something. A new diagnostic protocol that was approved this year is tomato brown rugose fruit virus, where ELISA is not recommended because of two reasons: analytical sensitivity, but also cross-reaction, so problem with analytical specificity. And so this performance criteria, this performance characteristic really help you to make a decision. And also for this protocol, and we know that it is also work in progress, but some of the PCR tests were also not included, not selected, because of cross-reaction with other viruses. And there are many more examples. So a recent diagnostic protocol we are working on, there are tests that are recommended on pure cultures only, but not on plants and seeds, because we don't have data on this type of matrices, and or the analytical sensitivity is not good enough. Philosticta citricarpa, it was demonstrated that the current tests are not specific enough to distinguish citricarpa from paracitricarpa. And so now, for the moment, this led us to a recommendation that the only way now to make the distinction between the two is culturing and sequencing. So this is also a use of validation data to say, well, this test is not good enough, so we need to to go through another route. We cannot only rely on PCR. So Erwinia, you can have symptoms that are really close to Erwinia amylovora, but are not Erwinia amylovora. So you need to be able to distinguish them if you go through testing. So all this is really also stressing the importance, and this is something we are repeating here in EPPO, that there should be really communication between diagnosticians and risk managers on the test selection. It depends what the risk managers expect from the test, and it's really important that there is this discussion between the managers. And there is another webinar from that will take place in February to tackle this question on why communication of test selection between risk managers and diagnostic laboratories important. And with that, that takes me to the end of this first theoretical presentation. So as explained, there will be a very practical session on the test selection and how you use a performance characteristic to select the tests. So do you have any questions? So Charlotte,
Yes.
I'll hand it back to you.
Yes, so thank you very much for the very nice presentation. So if you have any question, there is two ways to answer the questions here. Either you put your question in the chat, or you can also raise your hand and we will give you the floor. So there is one question in the chat, Françoise.
Yeah, the sensitivity. I'm probably getting confused all the time. So the level of pest you detect. So
maybe you can say the question. So just the
yeah.
question.
Oh, sorry. So the question is, okay, I need to try to enlarge it. So is more sensitive test the same as low sensitivity? I would think that it was the other way around, more sensitive, high sensitivity. So if the test, what I want to say, and probably I express it wrongly, is the quantity, the lower the quantity of your target can be detected, the more sensitive your test is. Is that clearer now? Is the person who wrote the question in the chat?
Yes, he said it's clear.
Yeah, okay.
So
I'm sorry that it's not the first time I'm a little bit confusing about this.
So then there is another question regarding the example of Xylella fastidiosa. Is it possible to expand the reasoning for the fact that one single ELISA test is acceptable in areas with known outbreaks and not a more sensitive test followed by a direct test?
Well, it's not telling you yet that you should use ELISA. It's telling you that you may use ELISA, but you don't have to use ELISA. I know in many areas that the more sensitive test is used as a start. So, or I hope I didn't misunderstand the question. So I'm
Maybe
trying.
the question was, is it possible to extend the reasoning to other pests?
To other pests?
Yeah, I guess it was this.
Okay, I'm trying. Okay, so now I can see the questions. So regarding the example, is it possible to expand the reasoning for the fact that one single ELISA and sensitive test? Well, I don't understand the more sensitive test followed by a direct test. So the reasoning, if I can explain the reasoning, it was that the analytical sensitivity of ELISA would be okay in areas you would be okay in detecting the bacterium in areas where it is present. So, but we have had a lot of discussion on that. So I can see you're continuing. Why more sensitive tests followed by? I'm sorry. I'm just trying to. I'm just trying to follow the question by Chantra.
But I guess it depends on the pest, and you will always
Yeah,
have
it
to
depends on the
put
pest,
it
but
in
indeed,
context.
in this case, in the context, I would recommend that you go to the PM4 PM724 standard, because here we've tried to explain this reasoning, and if you still have question, then you can come back to us directly, and we will try to answer with the help of the Xylella drafting team. Okay, so then I have a question from Quentin. Is there a guideline for morphological identification? So you mean for validation? So the guidelines are for the different disciplines, and it can include morphological identification. So the next one is, are the webinars recorded? Will they be sent around to participants? Yes, they are recorded. Charlotte, you can answer this one.
Yes, they are recorded, and they will be, the link will be provided to the participants and also on the website.
There are some questions that I'm not sure. So can one validate one's test only through EURL organized PTs? I'm afraid I don't understand the question.
I think the question is it enough to participate to a PTs to consider that you have validated the test in your lab?
PT is performance of the lab, so that's not validation. The PDF handouts will be available.
Yeah.
Let's cross-react with other species. So Simona, I'm working on diagnosis of a plant pathogen, and we are having problems in exclusivity. The test cross-react with other species in the genus, but are not related to the matrix as well. So your question is, you want, so you having cross reaction with species that you will never find in the matrix? Is that what you mean, Simona? Yes.
Maybe Simona, you can take the floor. I will unmute you.
Do you hear me?
Yes.
Yes, we hear you.
Okay. Our problem is that these species in the genus are not related with the matrix. So by now we are performing non-target PCR and qPCR, but the problem is inside of the genus. And by now, so far, not of the species tested in the genus are associated with the matrix. So
I think in this case, you can, when you do your validation, I'm not a lab person, so it's really too bad that René is not there. But for this type of question, but I would say that if these are species that are never related to the matrix you're tested, it shows you that there is a potential for cross reaction, but it's unlikely to happen in the real testing.
Yes, my question was, maybe it could be reasonable to continue to develop this kind of test, because obviously they are by now not related with the metrics.
As long as they are not related to the matrix, I would tend to think that you can still use them.
Okay.
Okay, I can see a lot of questions that are more for real diagnostician, which I'm not. So I don't know if the people let me check.
So there is, yeah, maybe you can check by
I can
yourself.
check the different, so allows much more material
There,
to be tested, and yeah. So Monica, so do I miss other people because it's
no, I think after next question is Monica's question.
Yes, so Monica, of course ELISA is more high throughput, but so far the problem, as I explained, is more about the cross reaction. And also, if you have symptomatic material, the drafting group considered that it's a possibility to use ELISA, but at the moment, the drafting group didn't feel comfortable adding ELISA for tomato brown rugose fruit virus. But I am conscious, Monica, that ELISA is you can do more material for sure. Yeah, PT
So,
is proficiency test. That was YACA.
yeah.
We know that it's proficiency, but yeah, it was. We're not sure I understand. To understand the question. So Anton, you had a question about what about viable reference strains but not culturable? It is possible. What do you mean by it is possible to use for validation? Anton, do you want to have the floor?
I'm looking for Anton, sorry.
Can I give him the floor? Or?
Yeah. If you saw him, yeah.
I saw him, but on the question, so not on
yeah,
the.
That's all right. Okay.
Yes.
Okay. All right. Yes. Françoise.
Good
I have a
afternoon,
problem.
Anton.
Hi.
Yes.
Bonsoir. Bonjour d'abord. Yes. The problem is that about reference strains. As you know, the laboratory is not yet equipped. My laboratory. And how come I have to do the validation of tests when I cannot use the reference strains, the appropriate reference strains? Is it possible to use, let's say, non-culturable strains?
So you mean how do you call it? The lab people would know better. Deactivated. I think you can use deactivated strains, but then you cannot keep them.
Oh.
You cannot keep them, but you can have them as reference material. But then that again, that's more a question for people in the lab. So if some, I don't know if in the attendance there were a number of people who were from labs, I could see that. So if they want to also contribute by giving answers. So
perfect. All right.
Esther, that's a little bit outside the scope of this webinar, your question on the flexible scope, but there is no definitely, I would say there are differences between accreditation bodies regarding flexible scope. That's a reality. We're trying to work on it, but it's a reality that there are differences, clearly. Okay, so how frequently happen that already validated tests are validated again? It's a tricky question because validation is very is validated for what? So you could have validation data in a publication, for example, and when you do your risk analysis, you see that some strains are missing, not all the hosts have been evaluated. So you could also try to adapt the test to test on different hosts. So yes, it happens, but how frequently? I don't have any idea. Regarding beside the sensitivity performance, would you recommend the combination of serological and molecular tests? We usually, in diagnostic protocol, it is usually recommended to use tests based on different biological principles. So yes, this is kind of a horizontal general requirements that to have a positive result, you used different tests based on different biological principles. When changing the chemistry for molecular tests, primers as used from. So that's a question from Marek Woźny. So if you change something, if you change chemistry for a molecular test, I would recommend, I think you should do a verification first, and please check the standard on how to do it. If everything is explained there, when you make some change, the first step is to do a verification. If you think the change are not critical, you do a verification to confirm that it performs equally. If this is not the case, then you will have to do the validation. And not only to test the sensitivity. That would be my but again, I would tell you to go again to the standard, and everything is explained. So reformulation of the two questions. Will an extremely sensitive test able to individuate Xylella way before its apparent use? Okay, that's.
Jonathan asked for the floor, so maybe he can ask
Okay,
the
yes,
question.
please.
I
Yeah.
will mute him. Oh, I'm not sure it worked.
Can you hear me?
Yes, we can hear you.
Well, actually, I asked before for the floor because I wasn't able to find the chat only. I'm sorry. Hi, I'm Jonathan from Israel. Actually, I raised two questions about the whether it can be useful in the case of Xylella having an extremely fast detection of Xylella, even where it doesn't appear yet, in order to allow very early phytosanitary intervention. I mean, as a result of regular periodic monitoring. And the second question, whether there is an advantage in this case in having a test that individuate only CFU, only live viable microorganism. I'm asking that because, correct me please if I'm wrong, I understand that PCR cannot really distinguish between live and dead bacteria or microorganisms.
Yeah, clearly.
Yeah, so I'm trying to understand whether there will be an advantage, a value to that.
Well, the quick detection is the advantage. So, and then the first time you have an outbreak, I know that Xylella is a difficult bacteria to culture, but you should try to culture it. So I think most of the having a quick detection test is also to take a quick action, and then there's always a decision to be made. So then we're coming to the discussion between the manager, the risk manager, and the laboratory. This is my test result. And then the manager will have to decide based on the sensitivity on the test, the likelihood that the pest is the consequences for the country as well. I mean, when you find a pest for the first time, it's a decision of the manager to know if they want to take an action or not. But we're beyond the topic of validation here.
Yeah, sure. Just may I ask how long it takes today to have a reliable result in how long it takes today for Xylella?
With what?
To have a result for detection of Xylella and enumeration of Xylella in the sample, how long it takes today?
That's a question to ask to the laboratory.
Okay.
EPPO is not a laboratory.
Okay, thank you very much.
Real-time PCR is very quick. Is less than a day. If I'm not, I don't. But it's you can have a result very quickly.
I assume a day after cultivation. Anyway, thank you very much.
The cultivation is different
After
because
cultivation.
it's a slow bacteria and it's not so easy to cultivate. So, but then again, go to the database on diagnostic expertise and you will find experts on Xylella there that can give you a much better answer than I can.
Thank you. I appreciated that. And thank you very much for this webinar. Thank you.
Yeah.
So Françoise, we are quite past the time. So maybe you can just answer the last question from Mirella. And maybe I think that Chantra we can see directly by email maybe.
Yes, we will send. And Lina, the standard is not yet published. It's in press. So we are doing the publication. But if you want a copy, it's no problem to send it to you. So I think I have your email, but if not, just send us an email and we will send you the version. We can consider the EPPO TRIPS PALMI as validated tests. So if they are validation data, not in some tests, in some diagnostic protocol, there is no validation when it's not available. We try to have validation and it's clearly mentioned when it's the case. So if validation data is present in the PALMI protocol, then you can verify that that's enough. So you can, and then the last one, you mean Chantra? No, we will answer.
Chantra said, yeah, I think it's okay.
But because you're not looking, the standard, sorry, I wasn't clear. It's PM724, not 84.
Yeah.
So, and don't hesitate to send an email if you want explanation. Okay.
So thank you everybody for attending this webinar. So next webinar will be next Friday, and this webinar will be how to adopt a new test in your laboratory. So thank you very much, and see you maybe on Friday. Bye-bye.
Bye-bye.
Au revoir.
Au revoir.